And there is our next guest. Well, thank you for joining us here on Giving Tuesday. I really appreciate your time, and tell us a little bit about yourself and the work that you do.
Okay. Okay. Thank you, Tim. Thanks for having me here. I am Yi-Wen Chen. I'm a principal investigator at the Children's National Research Institute. And our department also affiliated with George Washington University. So I'm also a faculty at the George Washington University. Wow. So I have been studying FSHD for about 20 years. Early on, we really focused our research on finding the genetic cause and also the disease mechanisms of the disease. More recently, we focus on therapeutic development and studies that are important to clinical trials, such as biomarker studies that can be used to evaluate the treatment, the efficacy of the treatment. And also we're trying to develop improved diagnostic methods that is more effective and affordable. So these are the few things we are really focusing on top of understanding more of the disease mechanism. So one of the major focus is the treatment that we're doing. And as you know, and I believe most of you know very well that DUX4 is a protein that's only made in a very specific type of cells at a very specific time during a specific period of time. Such as during the very early on in embryo development and in male germ cells. So after the DUX4 complete its job, our cells basically stop making these protein, DUX4 protein. So when the DUX4 protein is made in a way that's we don't we don't expect it. So basically at the wrong place at the wrong time, then they actually can interfere with the normal cell function and even kill the cells. So that's what the FSHD researchers believe what's causing the FSHD. So when we think about the DUX4 is the culprit, my laboratory, what we are doing now is actually to study several methods to reduce DUX4. And most recently, we actually collaborate with Dr. Toshifumi Yokota at the University of Alberta. And we developed an approach called GAPMER, antisense oligonucleotides. And this is a small stretch of DNA that actually can seek out the specific DUX4 mRNA inside the cells. So as you know that the mRNA is a copy of cell containing the genetic codes that's used to make protein that's made in our cells. So the GAPMER basically just target the specific DUX4 mRNA, bind them, and this process will actually activate a normal cell response inside our cells, and then our cells will actually degrade the DUX4 target RNA specifically. So that's the principle of the treatment that we are working on at this point.
Wow.
Yeah.
I mean, that's just that's like science fiction, right? I mean, that, but it's not. So the fact that it's going in there and attacking the DUX4, targeting it that in such a way, what's kind of the biggest hurdles or struggles that you're coming across, trying to make that science fiction sound, reality?
Yeah. So whenever is a great question. So whenever people try to develop an antisense targeting a specific mRNA that produce a gene product like DUX4 protein in this case, the mechanism, this mechanism in the cell is, like I mentioned, is a natural response. But the antisense that doesn't last long, that was a major challenge. And also you want to make sure these antisense can get delivered into the cells. And you want to make sure it's safe in the whole design. So there are many modifications that people use to stabilize the antisense to make it last much longer, from a few minutes if it's in its natural form, to now it can last for weeks or even months. So that's actually one of the challenges people have been really working on. The researchers has been working on in this field. And the other thing is once you do that, but then you still want it to be able to tightly bind to the target, so it doesn't bind to other sequences or other genes. So there are additional modifications researchers has been doing, and to increase affinity and specificity of the treatment. And then the other thing about the gapmer we're working on is we did some cell studies, and then we did animal studies to show that this is a compound that you don't need additional carriers to actually deliver it. So it can get into the muscle directly. So these are the different things.
We had a guest on earlier, and they mentioned, taking like a virus, and that virus would carry it into the muscle. So you're saying that this possibly doesn't need that.
Right. So a lot of other technologies similar to this will need either a viral vector or conjugated with a protein or some other molecules that actually help to deliver. This antisense, they don't need that. They can get in directly. Of course, if we can conjugate with a specific other things that can actually take them specifically in, that probably would be even better, because you can use a smaller amount of the drug and maybe deliver more specifically. But at this point, it doesn't need to be prepared in combination with another methods to deliver.
Fascinating. I'm learning a lot today, by the way, Yi-Wen, believe me. And so this is like the best science class I've ever had. So what is the possible, projected timeline, to have therapies get into a clinical trial state, do you think?
For this one, we have been finished. Usually when you develop a drug, there's a process, right? So you start with testing its efficacy using a cell model, and we use we actually use the cells donated by patients. So thank you for all the donors that donated the cells, as well as their family members, because we also need a control that show us this is what expected when they are healthy, the healthy cells. So we have done that. We show that they can easily get in and at very high potency. We did the animal studies more recently to show that we can do subcutaneous injection, and then the drug will get into the muscle, effectively reduce DUX4 to the extent that the animal can recover its muscle function and pathology. So, and then without obvious safety concerns. So we did the initial toxicity screening. We didn't see any safety concern. So the next big thing for us to do is really to do a full-blown GLP is called good laboratory practice toxicity study. With the smaller animal models and also large animal models. That's what people usually do. So we can collect data for FDA IND application, investigating new drug application. Once that's done, we will be able to bring it to clinical trial. So as you probably know that we are at the stage that need a lot of funding because other studies are quite expensive, including the toxicity and large animal studies, and move on to clinical studies. So we are really working hard to try to identify partners to really work on this with us, because we are entering a stage is no longer be easily supported by grants. And We do have several companies that show interest, including venture company. So we're really, so I think the answer is if we actually can get this set up quickly,
Yeah.
identify funds, then it can be really quick.
Wow.
But that's is.
So it kind of needs that foundation of funds, the back end, you said, of a company. Sounds like, community support is something that we're actually trying to, create a little bit here, with Giving Tuesday, and trying to, we got matches of our dollar per dollar, all the way up to 150,000 today, and that kind of impact. So I assume then community impact, not just for donation, as you talked about, donating their cells, but also dollars is pretty important. You want to speak about that?
Definitely.
Yeah.
I definitely want to talk about this because I have to say that when we started this project, it was an idea. And without the support from the society, we actually received an initial fund from the FSHD Society that actually helped us to launch the study. That's how we complete our cell study and the initial animal study. So there's no way we can really materialize this idea without the support. And like we just talked about the donors for the cells. And there is also another study we're conducting, we have been doing. It started a while ago as a clinical study that's actually studying the early infantile form of FSHD. And that was also started with a seed fund from the FSHD Society. Without that funding, we wouldn't be able to start this clinical study, collect clinical data, and as well as the blood samples from lots of participants in the study. Again, that's another way to contribute. There were participants who are affected by FSHD. There are also participants who are their family and friends who donated their blood samples in this case. So we were able to actually compare what's in the patient's blood and what's in the control healthy individual's blood, and actually identify, that's a paper we just recently published, identify protein and microRNAs that's different between these two population of individuals. And these are supposedly very good circulating biomarker candidates. So the goal is actually for us to identify a group of them so we can actually take blood samples and then examine the values of these potential biomarkers and then determine whether a treatment is effective or not. But this is really a long way from the beginning, was just characterization studies, which was funded by FSHD Society.
And you've been, because you said you've been doing this for 20 years, investing your time, which I'm beyond grateful for that kind of commitment, and that you continue to press ahead, because I'm sure it's been a long road for the 20 years of your research work. What's kind of the biggest change between when you started to where it is now?
I think there's a lot of changes. Of course, the size of the community, the number of people join the society and actually put in the work, it definitely is growing amazingly every year. But in addition to these changes, I also want to say something that's really not changed. I think the support from the society has been really, supportive from the beginning to the scientific community and also the patient community. My first scientific meeting about FSHD was the meeting that's hosted by the society. That's where I met a lot of scientists that's studying FSHD at that time and now, that really form all these collaborations and then speed up the process. I think that's really important. And in the process, I also get to know a lot of individuals affected by the diseases. And I'm always amazed by the energy and all the impressive, how much they devoted to the society and the cause. So I think this is a great community. I have to say that definitely it grows a lot. So a lot of hard work and wonderful work done, by June and Jamshed, Mark, and other, staff at the society. But there is also something that's been there always, and I'm very grateful of the society too. They are doing a great job, and I think it's really great.
If people want to learn more about what you're doing, is there a place that they can look, or maybe better specific, do you need more help from the community, not just financially, but also donating cells, or more of the involvement from the patient community? And how, if that is the case, how can they help?
Yeah, definitely. I think the patient community has been doing a great job, but definitely in addition to donate to the donate money, so additional exciting scientific project can start or continue, or even just to uh, help to facilitate to actually recruit more young scientists into the field. These donations are definitely necessary for just moving this whole thing forward. There's no way we can do it without participation from our patients and their family members, because when you started a human trial, you will need participants to get these done. And we talk about the cell models. That's pretty much the most critical first step if someone wants to test a treatment. So that's definitely helpful. I would say anything a person can do. And I think the FSHD Society is doing a great job at on coordinating all these efforts. So I would say probably become a member and join the society, and then see what's going on, and then do whatever you can do whenever it's needed.
That is great words of wisdom, and we appreciate your time today, Yi-Wen, for joining us and talking today about research. I've learned a lot more since our conversation started, so I greatly appreciate your time today.
Thank you. Thank you for having me.
You bet. That is Yi-Wen Chen. She is a researcher for the Children's National Research Institute.